Somatic cells are all body cells except reproductive cells. They have 46 chromosomes and are created by mitosis. Gametes are cells used for reproduction. They have 23 chromosomes and are generated by germ cells through meiosis. The cell division cycle is the reproduction of cells. The G1 phase is the cell growing by producing proteins in cytoplasmic organelles. The G1 checkpoint is checking to see if the cell is big enough or if the DNA is damaged. The S phase is DNA synthesis, where the DNA is replicated. The G2 phase is the cell getting ready to divide. The checkpoint is checking to see if the DNA is correctly replicated or damaged. The G0 phase is where cells don't divide, but have completed the G2 phase.
The first phase of mitosis is prophase, where chromatin fibers begin to condense into chromatid and appear as sister chromatids. The spindle begins to form, composed of chromosomes and microtubules, and DNA strands are wrapped around histones. Next is the prometaphase, where further fragmentation brings about the breaking down of the nuclear envelope, and centromere is connected to the kinetochore of each of the sister chromatids. In metaphase, centrosomes are now at opposite poles of the cell, and chromosomes centromeres line up on the metaphase plate. Microtubules attach to the kinetochores. In anaphase, the kinetochore microtubules shorten, so two daughter chromosomes pulled to opposite poles of the cell, and the cell elongates. In telophase, there are equal chromosomes at each pole. Two daughter nuclei form, and the nuclear envelope arises, and the chromosomes are less condensed. Next is cytokinesis, where the formation of a cleavage furrow pinches to the cell in two in animal cells, and in plant cells, vesicles fuse to the cell plate to form a new cell wall.
A diploid cell is a cell with two sets of chromosomes, represented by 2n. In humans, 2n equals 46. The first stage of meiosis, which is how germ cells reproduce to form gametes, is prophase 1. During prophase 1, homologous chromosomes, which are sets of chromosomes from both the father and the mother, join together. This is called synapsis, during which the paired homologs become physically connected to each other. During synapsis, crossing over occurs. Crossing over is the genetic rearrangement between non-sister chromatids in which they exchange segments of DNA molecules. This occurs at the chiasmata. Following prophase 1 is metaphase 1. After metaphase 1 is anaphase 1, during which the breakdown of proteins allows for homologs to separate. Spindles guide homologs to opposing poles. During meiosis 1, independent assortment occurs. Independent assortment is when each pair of homologous chromosomes sorts maternal and paternal homologs into daughter cells independent of every other pair. Meiosis 1 results in two haploid cells with two sister chromatids. The next stage is meiosis 2. Meiosis 2 begins with two sister chromatids of each chromosome that are not genetically identical because crossing over occurred. These chromatids separate and move towards opposing poles, where the chromatin decondenses. The division of the parent cell results in 4 daughter cells with unduplicated sets of chromosomes. These 4 daughter cells are genetically distinct from the parent cells and from each other.
Review! Mitosis is done by somatic cells, which is the cell division resulting in 2 identical diploid cells. Meiosis is completed by gamete cells and occurs from germ cells, which make 4 genetically different haploid daughter cells. Thank you!